But yeah, so I'll just briefly, if you don't know me, I'm Deidre Jansson, junior faculty with Jeff's group, and this is the basic biology that we're interested in. It's like the kind of the science that our whole lab revolves around. So it involves the anatomical distribution of perivascular pathways that allow cerebrospinal fluid to exchange with interstitial fluid, kind of facilitating movement, distribution, and clearance of different kinds of brain solutes. So there's a lot of different, I guess, parts of this biology. And so we work with animal models, we do a little bit of in vitro work, but also like just having human histology is it's just this amazing resource so we can actually ask human relevant questions about this biology. Um, so if the first, yeah, so I just want to talk about, and Caitlin outlined this, so I don't even need the slide, but it's just this amazing rich data set that has like a plethora of tissue samples, like paraffin embedded or frozen, and like the cell lines. And this is, I guess, coming from a small lab in New Zealand that was claimed to be world famous for our brain bank. It seems sort of funny to think about that now because we had, a few hundred samples and it was sort of, you had to like fight to the death to be able to get samples to actually ask your science question because everybody wanted them and they were so limited. Um, and I think this has just been a completely different experience, very refreshing in that I felt so supported, I think, in like the questions that I wanted to ask and just getting help. And like if I, didn't know, obviously Caitlin is just like this amazing expert. So providing that expertise and like understanding the the human specific questions that you want to be asking in terms of when you're like what kind of tissue you're going to get. So I'll just go through a couple of examples of how we've done this. So I'm personally maybe the only one in the lab really interested in the choroid plexus. So it's a highly vascularized tissue that sits inside the ventricles, secretes cerebrospinal fluid, and has been implicated in like a number of different inflammatory diseases, but Alzheimer's disease is one of them, as well as just aging. So there's a lot of changes that occur. And the evidence from this is provided from some animal models as well as some human sort of postmortem tissue samples. Um, and I guess the questions that I have are more related to like the cell specificity of it. So there's different cells within the choroid plexus, the epithelial that line the outside. It's highly stromal, so there's a lot of fibroblasts. There's endothelial cells. There's pericytes, and it's highly inhabited by immune cells. So macrophages, there's T cells, B cells, and then they're even able to cross that epithelial border depending on the circumstance and like the inflammatory condition. Um, so yeah, so like I said, it's potentially interesting. It might be involved in disease processes. So if you think if it's making the CSF, maybe it's contributing to distributing like inflammatory molecules. It's also known to be involved in like taking up A beta. Um, but I guess the data so far was a bit limited because we had like some animal models and like that quadriplexus is very different. It's a lot simplified compared to human. So I wanted to ask questions about human choroid plexus. Which cells are potentially damaged? Which ones are the most changed in the context of Alzheimer's disease? And again, started out like very naive, and I was like, oh, just Alzheimer's disease. So I really had to specify exactly what I wanted to be looking at in terms of, the pathology, like the Braak staging, like how much tau, how much amyloid. So we applied for some funding through the ADRC development process and to perform a pilot study to do single nuclear RNA sequencing of human choroid plexus so that we could hopefully look at individual cell contributions, or I guess their roles in the context of Alzheimer's disease. And since we were able to, I guess, get the pathology data. So in terms of Braak staging, fall, even arteriolosclerosis, age, sex, ApoE status, that we could ask a lot of questions about like specifically epithelial cells with ApoE4 kind of express these genes more. So anyway, so I'll go into that. Um, this is just the basic things that you see when you do single cell um, science. So we were able to get samples. In the end, I think we had 9 controls and 9 clinically designated Alzheimer's disease. And so we needed fresh frozen tissue. And when we got the samples, we didn't actually have all the data, so we were just dividing it based on like clinical Alzheimer's disease versus controls. Um, and this is what we got. So we got a lot of cell types that we expect: epithelial cells, mesenchymal cells. There's some immune cell types. There's some endothelial cells. So it's looking really good. And then I guess later on down the line, after we've done the single cell part of it, we got the path data. So we could actually like kind of put that data into our analysis pipeline. So the Braak staging and thal and vascular disease characteristics. So if you can see these, so we can essentially like split up our clusters just based on all those characters I just mentioned, and kind of see how the distribution looks like. Again, this is like a minimal amount of samples, but it's looking pretty good. And then the next thing we do is look at the different genes that are most differentially expressed in the different cell clusters. So the cell clusters are down at the bottom. They're annotated by color. It's just a heat map looking at the distribution across all of them. And then we do, again, it's very, very general, like RNA-seq pipelines, pathway analysis. So what types of biological functions are most represented by these genes that are changed between the different cell groups. So we can sort of answer more specific questions about, I guess, function in terms of related to. So I'm just giving you an example. So this is with Braak. So with like higher BRAC, there's pathways that are upregulated that are involved in wound healing, hypoxia, um, lipo lipopolysaccharide. So that's like inflammatory mediated cell substrate adhesion. So this one at the top, 0, these are epithelial cells. So there's the ones the CSF facing essentially, and they have like a blood CSF barrier. So it looks like there's some sort of barrier properties that are lost. And again, this is in line with generally what we understand that from just like human histology studies and some mouse models. Then we do the same thing, but now we put in thal to the algorithm. So what does it look like when we compare genes across low thal versus high thal? And yeah, we get, I guess, essentially like sort of different functions that are associated with more tau. So with more tau in the brain, epithelial cells seem to change their regulation of ion transport, membrane transport, calcium homeostasis. So um, I'm quite excited about this data set. I know it's just a pilot study, but because we've done it, and I guess when we started out, this was going to be the first one, but we're now collaborating with a group in Boston that also has a single cell choreplexus data set. So we're trying to, yeah, like look across these data sets, see whether we can, one, combine them, but if we can't, maybe just looked at consistencies across them. And I was able to do this actually because when I started in Jeff's lab in 2019, before I even got to Seattle, I got in touch with the Neuropath core and I said, hey, I'm really interested in choreplexus. Like, do you guys have these samples? And so they started collecting it for me. So I literally have hundreds of samples, and I have pathology data like BRAC and thal and age and sex, a lot of them ApoE. And it's just like this, I'm almost certain that we are the only people in the world that have this. So it's just like, it's kind of like this selling thing that I have, like, please, like, come collaborate with me because we have this amazing resource that's been provided. So a really great thing has come out of that. So I'm really excited. And then, yeah, so I'll move on to the next one. Unless anyone has questions, it's probably easier to just go at the end and you can, yeah. So that's that one that I'm really excited about. And then this next study I want to talk about is the one that I'm working closely with Caitlin, as well as it's funded by the Leduc Foundation for Brain Clearance and Cerebral Amyloid Angiopathy. This is a lot newer, and so there are, I guess, there's evidence to suggest that there's impairment of clearance in the context of CAA. But essentially, like nothing's known about really whether it's something that drives disease processes or it's sort of just an later effect of amyloid deposition within the vessels. But um, to give you a brief background, CAA is usually clinically like sort of classified, not clinically, but classified by stages, with early deposition of amyloid around the vasculature, typically starting in the meninges. Um, and then subsequent loss of vasoreactivity with more amyloid deposition and then smooth muscle cell loss. And that leads to microbleeds and hemorrhagic injuries and eventual death. So yeah, the goal of the Leduc Foundation, the consortium, is to, um, I guess, answer questions about clearance in CAA. So what we did, again, because recognizing that we've got this amazing resource with the Neuropath Core, reached out, to Caitlin and say, is there something that, can we do something with the tissue samples that are available? And so, yeah, Caitlin looked through, and there were enough cases with CAA. Again, so sorry, I'll back it up a bit. So, like Caitlin mentioned, you can't just ask for like CAA or not CAA. A lot of Alzheimer's disease cases have CAA. A lot of CAA cases have amyloid plaques. So eventually we ended up asking our question in the context of CAA, either with amyloid plaques or without, or hopefully like amyloid pathology with low CAA and versus healthy age-matched controls. And what we wanted to ask about, and this is reminding me that I didn't even mention aquaporin 4, but if you've heard me talk before, you've heard it. But essentially, aquaporin 4 is a water channel that's expressed at the astrocyte end feet that line the perivascular spaces of the blood vessels. And it plays a role, so at least in animal models, and there's pretty good evidence to suggest that it's involved in human disease as well by pathology, like human path staining. Um, so we wanted to ask the question of whether aquaporin 4 um, has any sort of association with cerebral amyloid angiopathy, whether it's region specific, whether it's associated with inflammation, or like the presence or absence of amyloid plaques. So yeah, so Caitlin found these tissue samples that match our criteria. So this is what we're starting with. And this image on the side is a beautiful image of red lectin stained vessels. And the green, if you can see it, is amyloid, and then the blue is aquaporin 4. So what we want to do is essentially quantify this aquaporin 4 staining around the vessels. And so how we're doing this, because the tissue samples are so great, they just look so pretty in these pictures. We're using AI, deep learning, and artificial intelligence deep learning analysis to do this. So it's a VisioFarm platform that we're teaching to recognize gray matter and white matter regions, as well as the meninges. And then this is just an image of sort of what you do. You go through and you train it and say, this is white matter, this is gray matter, and then you test it out to see if it's working. It's doing pretty well. We're still working on optimizing it. And then what we do is use like a thresholding approach to make almost like a mask of the red lectin vessels, and then picking up the green, and then being able to differentiate between amyloid positive versus amyloid negative vessels. And then once it does that, it will make them like amyloid positive vessel like an object, and then we can draw these concentric rings around it, and then it can quantify the staining within that region, which is aquaporin 4. So that's essentially where we're at with that one. But yeah, I'm really excited. The images look really great. And I think just because we have the information on like BRAC and thal, but we can also do quantification of amyloid within these tissue samples as well. We can really get nice, pretty, I think, clear-cut relationship between aquaporin 4 expression and amyloid. And then this is my fantasy experiment. This is not my experiment. This is something that Caitlin and Angela have run using DSP to quantify protein expression around either amyloid positive or amyloid negative vessels. And so ultimately, after we do this really nice, hopefully like very high throughput analysis of like per vessel aquaporin 4 expression, we can go back and then do this and essentially ask what proteins are associated with high perivascular aquaporin 4 or low, and in the context of amyloid or not. So those are that's just examples of the stuff that I'm working on with the Neuropath core. I've also done kind of what Jeff had mentioned, like I'm a mouse person, I need to validate in humans. So I have another project that was in mice and in vitro, and I need to validate with some human stroke tissue. So I requested that, and that was provided just so that we could validate with some RNA-scope experiments. I'm also working on a study with Swati Rane Lewandowski, which is she's works at the MRI uh, Center and the UW, and we are asking questions about perivascular spaces that are usually enlarged in the context of like CAA as well as sort of inflammatory disease. And we're matching MRI data to the histopathology and the perivascular spaces that we see in tissue. So I didn't go through that because it might be too much, but yeah, that's pretty much it. And again, it's super easy. It's a really supportive environment to ask really great science questions, and I'm happy to answer any questions, or if anyone wants to email me later, you can do that too. These are just some of the people I'm working with in my email if you need.
Good, thanks, Dee. We have about 10 minutes. If folks have questions for Dee or more questions for Caitlin, now is your chance. Maybe Dee, you could, I'll start with one. What was the biggest surprise that you encountered in making the transition from someone who worked with rodents or worked in model systems and things like that to working in real human populations? Like what were some hard things that were difficult to learn, or where you had to change things quite a bit?
I guess I would say just the variability in humans in general. Coming from even I think even the like brain bank that we had in New Zealand, we didn't get nearly as much like clinical or pathological data on the samples. We were just kind of like, this is an AD case or this is a Parkinson's disease case, and that's really all we had. So I guess just recognizing and getting a little bit of like statistical support as well to actually make sure you ask the right question. Like you really can't just say Alzheimer's versus controls because like Caitlin said, you don't find controls without plaques really. Like it's very rare, especially like with older people and with older people. I think that was sort of the biggest, I guess, like new thing that I came across. It was quite a challenge just to try to wrap my head around like how do we ask these questions about disease if kind of everybody has a little bit of disease.
All right. Other questions?
I wanted to just highlight one thing that Deirde brought up, which was generating a resource that probably not many people have, this choroid plexus resource. And it was not something we were doing. And so this has happened multiple times, and it's something we're willing to do kind of, on this piloty basis, depending what it is. I mean, so if we don't have what you need, if you need tissue collected in a special medium so you can test something out, that's going to require our autopsy techs when they're here in the middle of the night doing a rapid to change what they're doing. You know, we are willing to do that, usually on a small number of cases, to generate preliminary data, with the idea being, yeah, if this works, we're going to write a grant on this and do this. And we have collaborators like out at MIT who had a very complicated protocol, but now we're, you know, on a big grant that hopefully will be funded with them. And so we'll do stuff like that. We can add samples. What Deed wanted was pretty simple for us to do. The chord plexus is there, and it was just getting fixed, kind of stuck in other samples. And so it was relatively easy for our techs to add a step at collection and at when we do the fixation step, to have both a fixed and frozen sample. So if you have a question and you want something collected in a different way, it's just to say it's worth reaching out and asking, because a lot of times, especially on a limited sample size, we're willing to work with you and try to get you what you need to generate that preliminary data.
Very good. All right. Any other things? Jason, you just show
Yeah.
yourself.
So I was wondering, like, if somebody requests, like, say, tissue for histology, what do they receive along with that? Do they get information, like demographic information? Is there like accompanying things that come with a tissue request?
Yeah, I think I'd have to double check with John and our team if this is automatic. You could definitely click on what you're asking for. You can click data and make sure you get the data, but we have no qualms about sending the standard Neuropath data set along with those, which includes, like I said, all our standard Neuropath variables, but then also some of this basic information about the donors, you know, all non-identifiable stuff. And then if you wanted something extra, you'd have to specifically ask for that. But that all comes through us unless it's not a data point we have. And then we'd have to help you reach out to the people who might have that.
And I would just sort of amplify just sort of a point, and I don't want to obligate Caitlin or Dirk or Amber beyond what they're capable of, but the value here isn't just, they're not just like a tissue supermarket where you like go in and get some tissue and walk out and go to your car and go home. The value is in the relationship that you can develop with the core that is collaborative, where they help to inform you about what they have that could be useful. And so you start a conversation with them, and through that conversation, figure out, well, what data do they have that could be useful here, and arrive at what you need, right? And to me, the form is like the thing you fill out at the start of that to start the conversation that eventually leads to the final product. So thinking of them as friends, as opposed to suppliers, is maybe not, they're not your friends, but you know what I mean? Think of it as a relational thing as opposed to a transactional thing might be useful. Juliana?
Awesome. Thank you. Great talks, Caitlin and Dee. I just have a quick question for Caitlin. You had some information about the number of samples that the Neuropath core has, and this is just a curiosity about how many new cases do you add, you know, on a yearly basis? How are those numbers regenerated, and how many new cases? I know you've mentioned a few times, you know, the techs coming in to collect. How often does that occur?
Yeah, so we've gotten very busy because Dr. Keene is very good at establishing a lot of connections. And so I think we're getting close to 250 new cases every year, which is why, just as a side note, when Dee said she had the samples but not the Neuropath data, it's not because we just decided not to give her that data. There can be anywhere from 3 months to a full year before a Neuropath case is signed out. So because we're dealing with so many cases and some are complex. Anyway, so that's what she eventually got that data. But yeah, so we get about 200 to 250 a year. I would say a lot of that now is coming in because of our work with the medical examiners and trying to generate a resource of quote neurotypical brains, some younger donors, these trauma related cases that are coming through as sort of tragic unexpected deaths, as opposed to our older donors that are part of these long-term studies. So that's a big chunk of it. We probably get like 25-ish Alzheimer's Center cases a year, and close to, I want to say 60 or maybe 40, I don't know, somewhere around like 50 maybe ACT cases a year, but it kind of depend on the year. And so hopefully, our control, it's the control cases and the really bad AD cases that tend to suffer from the sharing. And so that's the main reason why, if you come to us asking for, every brain region on every case we have, we're gonna talk you down. And it's not because we don't want to share. We just want to make sure we're judiciously giving you what you need to do what you need to do, but not depleting the resource.
Yeah. Very good. Anything else from anyone else?
Yeah, one more quick question. So, Caitlin, based on your understanding of Alzheimer's disease and your perspective from the inside on the brain biorepository, are there things that people are not requesting that you really wish people would request, or think that there's a lot of potential that so far no one seems to be asking about?
That's a really good question. And this is only because, like, who would think to ask for this, but because we've generated these imaging blocks and then we have all this imaging data, it would be great to be doing more work with neuroimagers who want to do neuroimaging, neuropath correlations. And right now it's, a few small groups kind of very much within our core group that are doing this, but it's probably because we know we have these samples, and I don't know how obvious that is. Some centers like I always point to the Ross map because they are a very productive and large core. They have a nice interface where you can really see everything that they have available. And so that's always been something in the back of my mind, like, what could we do to be more visible about what we have, and make that a little bit more clear for people to kind of browse, not to turn us into, the Amazon of tissue samples. Those relationships are really important, like Jeff mentioned. We don't want people to just be able to come in and like click on stuff and I want this and leave, but it can still be a way for people to see, to window shop, and then come meet with us and say, hey, I saw you have these things. So I guess it's, all these neuroimaging blocks that are really mostly just sitting there right now.